Now, in bacteriophage experiment, what we have done, that bacteriophage is a virus that attacks bacteria. When we grow the bacterium in a nutrient medium containing radioactive sulfur, then radioactive sulfur incorporates in the protein coat but not in DNA. But when bacteria was grown in a medium containing radioactive phosphorus, then it will incorporate into DNA but not in protein. Now, this is the procedure of the experiment. This is the bacteriophage which was grown in a radioactive medium which was labeled with radioactive sulfur, that is 16s35. Now, this bacteriophage attacked this bacterium E. coli. When bacteriophage attacked, only the DNA entered and the protein body, it was left outside. Means the protein coat will not enter and only the bacteriophage DNA it entered. DNA entered, DNA multiplied, it made several copies of itself. Proteins, they are left outside. DNA. Now, the DNA of bacteriophage, it will use the raw material from the cytoplasm of this Escherichia coli and it will make more bacteriophages, and when we will check them, so radioactive sulfur will not be present in these bacteriophages. क्योंकि अगर वो ट्रांसफ़र होता, प्रोटीन जेनेटिक मटेरियल होता, तो वही आगे ट्रांसफ़र जाता और जो नए बैक्टीरियोफेज बनते उनमें भी रेडियो एक्टिव सल्फर देखने को मिलता, बट नो रेडियो एक्टिव सल्फर 35 was detected in the new bacteriophages. Now, when the bacteria, they were grown in a culture medium containing radioactive phosphorus 32. Now again, अब ये जो DNA एंटर किया है, it is labeled with radioactive phosphorus. It has multiplied by replication, made several copies. Now they will synthesize their protein coats और जो नए bacteriophage बनेंगे, they will be now radioactive phosphorus 32 will be detected in these bacteriophages. So this experiment given by Hershey and Chase, it proves, it gives the conclusion that bacteria infected with viruses grown in sulfur 35, they had no radioactive proteins, but bacteria infected with viruses grown in phosphorus isotope, radioactive isotope of phosphorus, they had radioactive DNA, which concludes that DNA is the genetic material and DNA passes from viruses to bacteria. Now, this was the experiment which we have completed. Now, there are few questions. Properties of genetic material, that is DNA versus RNA. Here is a question. Name some viruses. This is the question. Name some viruses where RNA is the genetic material. This is the question and the answer is number 1, tobacco mosaic virus. Number 2, QB bacteriophage. These are the two viruses which have RNA as the genetic material. Now, question is, what are the two chemical differences between DNA and RNA? Two chemical differences between DNA and RNA we have done. The two differences are, number 1, let us see. Two chemical differences between DNA and RNA. First, the purines are adenine and guanine, and pyrimidines present are cytosine and thymine. But in RNA, purines are same, that is adenine and guanine, but pyrimidines, they are different, that is cytosine and uracil. Means the thymine is replaced by uracil. The second difference is the sugar present is deoxyribose with the molecular formula C5H10O4, and here the sugar present is ribose, that is C5H10O5. These are the two chemical differences between DNA and RNA.